Good afternoon, everyone, and thanks for attending, and thanks to the IRM organization for letting us presenting Vivet today. Who are we? Vivet is a clinical stage company developing liver-directed AAV-based gene therapy for rare metabolic disorders. Our 5 pillars: we have strong translational capabilities with our research team located in Spain, in Pamplona, doing all the early-stage clinical work from animal model development, design of the construct, molecular biology, as well as first in vitro and in vivo proof of concept. We have direct access there to animal facility in mice, but also in NHP, which is a key asset. The rest of our development team is located in Paris, as you can notice from my accent. We have, secondly, a diversified product pipeline. Our lead asset, VTX001, is today in the clinic in Wilson disease. This is the one I will do the main focus today. And 4 other assets which share in common targeting rare disease with high unmet medical need, but also limited competition. Third, we have an innovative and modular platform, basically willing to address what we consider today as some of the main limitations of AAV-based gene therapy, which are the sustainability of expression. We have a non-integrative approach on that regard, as well as the ability to administer AAV product in patients with neutralizing antibody. On the CMC side, we have decided from scratch not to invest into our plant. It's very costly, as most of you will know, but we rather rely on a robust partner, and we'll do a small focus on this one, on one of our partners, which is Pfizer. And last but not least, we have committed investors that you can all see here. From the beginning, they are solid and committed. And talking about investors, I would like to do a focus on Pfizer. We announced this deal with them in March 2019. This is for us clearly a highly strategic deal in the sense that Pfizer became a shareholder of the company. They took a 15% stake in Vivet, and they have the exclusive option to acquire the rest of Vivet based on the delivery of certain data from VTX001, certain clinical data from our lead asset. We even expanded the collaboration with them. We announced that in September 2020, when we decided to work with Pfizer on the manufacture of VTX001. And clearly here we can benefit from their striking force in manufacturing. They have announced at the same time that they have invested $500 million in their gene therapy manufacturing sites in North Carolina, especially in Sanford, which is giving us a very, I think, nice 2 days, I would say, force in terms for the phase 1-2, but as well as perspective for the phase 3 and for commercial, which is key. I mean, CMC remaining a bottleneck today for many of the developers. So now let me do a focus on Wilson disease. What is this disease? So it's a rare metabolic disorder affecting, according to the literature, 20,000 patients in US and Europe, estimating half of them being diagnosed. It's an autosomal recessive disorder affecting the ATP7B gene and resulting in dysregulated copper metabolism. And the ATP7B protein has actually, 2 main functions. One is to load copper into the bile, which is then excreted into the feces, which is the normal route of elimination of copper. And second function is to load copper onto ceruloplasmin, the ceruloplasmin protein, which transports copper as bound copper elsewhere into the body. If non-functioning, 2 main consequences. One, copper starts to accumulate first into the liver and then elsewhere into the body, especially in the brain. And copper is not transported as bound copper, but as free copper, which is known as toxic. They are standard of care today for Wilson disease. I mean, it's a rare disease, but has been characterized for several decades now. So 2 main standard of care. One is the copper chelators, which basically chelates copper in the blood and helps it pump it up out of the body through the urine. And zinc salt, which prevents copper absorption into the intestine. But as you can see here, which is what is reported in the literature, there are important non-compliance issues in more than half of the patients. A third of the patients experience side effects, especially neurological side effects, and the so-called paradoxical neurological worsening when initiating copper chelators. This European registry has also been showing that more than half of the patients approximately are not improving, actually, because half are improving, but the other half are not improving on their neurological manifestation. And up to a quarter of them are still evolving on their hepatological psychiatric, or neurological component, which are the three main clinical components of the disease, despite being on treatment. And the last point, highlight that they are, depending on the country, but up to 15% of the patients that undergo liver transplantation. But this is what we know basically from the literature. But what we decided to do at Vivet is to also rapidly, in the early days of the company, invest in analyzing healthcare databases. We are also looking at registry and so on locally, but also looking at national healthcare databases to try to understand who are really in the real world, these patients, and what is happening to them. And what we first found out and confirmed is the prevalence of the disease. So we identify more than 12,000 diagnosed patients in the US, 2,000 in France, 1,400 in Germany that are treated, so which gives numbers which are in the range of what is reported in literature. And actually slightly higher prevalence in Asia. We are today working on a Japanese database where we have identified 26,000 patients. And a South Korean paper from another group has been recently published and showing a prevalence of 5 to 100,000 persons. We also looked at the disease burden. This is not an easy disease to diagnose. There are multiple components from the hepatic, psychiatric, and neurological aspect. And as you can see here in the US, the cost just the year prior to diagnosis is 10 times higher than the general population, 16 times higher just the year post-diagnosis, knowing that this patient can be misdiagnosed for years. In France, we found a very high rate of hospitalization, having 3/4 of the patients being hospitalized during the first year post their diagnosis for an average of 4.5 days, and almost half of them still being hospitalized 10 years post-diagnosis. Significant burden in terms of disability levels. I will not enter into too much detail there. We have different stratification there, as well as long sick leaves during the first years post-diagnosis of almost a third of the year, basically. And this point, which I think is key also to understand the disease, and that has been shown in this South Korean paper, is that patients with neurological or psychiatric component are actually managed for this condition way before Wilson is even considered and diagnosed, and it can last for 1, 2, 3, 4, up to 4 years, which I think is key to understand that there is a clear need for disease awareness and an improvement of that. But all in all, across the globe, US, Asia, Europe, we found very convergent data, late diagnosis in all cases, 25 to 35 years of age. More than half of the patients are still showing significant clinical manifestation despite being under standard of care. Much more surprisingly, and we are digging into that with further analysis today, many patients are left untreated. It could be for access issues, but we're also digging for other potential issues, especially awareness. Premature mortality. We are talking here about 20 to 30 years younger than the life expectancy in developed countries, as well as confirming the level of liver transplantation in this patient. And now I would like to do a short focus on liver transplantation. And why doing so? Because basically what we will do with gene therapy is replacing the ATP7B protein and aiming at doing what a liver transplant can do, which is restoring the normal copper metabolism. And this is coming from a French study, a small study still, but showing that in severely affected neurological Wilson disease patients, that you can see here, liver transplantation has been able to revert their neurological symptoms very significantly from a general neurological scale, the modified ranking, while here it's a classification specific to Wilson, the UWDRS. You see that patients have been able to go from very severe handicap to become independent for their daily living. And I think it's key because clearly today's standard of care is not able to do that, and we think it's very encouraging to see that, and let's see what gene therapy can do on that regard. So now let me give you some Vivet data, and what are we willing to do? So if you remember the copper metabolism, you know copper is excreted through the feces, which you can clearly see here in patients that have been injected with radio-labeled copper, copper-64. So in healthy patients, heterozygous patients, which have no clinical manifestation, although there is a bit of lag time, most of the copper is eliminated through the feces. While in Wilson disease patients, whether they are asymptomatic, symptomatic, on treatment, off treatment, you see almost no excretion in the feces. And you actually see excretion into the urine, which is even exacerbated by copper chelator, by on treatment. They just pump it up and excrete it through the urine. So what we did is in our Wilson disease mice model, injecting at a different dose, we've shown a dose-dependent restoration of the copper excretion into the feces up to the wild-type level at the highest dose, as well as a restoration of ceruloplasmin activity. So again, playing and restoring the two main functions of copper metabolism, of the ATP7B, sorry, and therefore normal copper homeostasis, which is correctly excreting copper and correctly transporting copper. As a result, we have been able to normalize the level of copper in the liver, level of copper in the brain, and we have shown 100% survival. We have data up to more than a year now, but at 1 year in the treated group versus only 40% in the non-treated group. So what are we willing today to do? We are now in the clinic with the Gateway trial. It's a phase 1/2 clinical trial run in the US and Europe, currently enrolling patients. We will do a first check of their copper 64. We'll inject copper 64 at day 0, at baseline. Administer VTX801. We'll do a second check at 3 months, second check of copper 64 at 3 months, which will allow us to discriminate and distinguish between responders and non-responders. And I insist on the point there that responder will be taken off completely out of their background treatment. That's the objective. And a third check at 9 months, first to evaluate the sustainability of efficacy in the responder and potentially identify late responders. So all in all, we consider that we have an innovative biomarker demonstrating really mechanistically that we are restoring the copper metabolism, the normal copper metabolism. Ambitious readout, which is the discontinuation of copper. Again, we are not evaluating to decrease the amount of chelators by X% or whatever, which we think gene therapy should allow patients to get rid of their standard of care. I think it's key for demonstrating the true value of gene therapy, and a rapid readout with the first evaluation at 3 months. So now, just as a corporate highlights to sum up, as you understood, our lead asset is 801, which is now in the clinic. We will announce first readout in the second half of 2022. We have a second most advanced program in PEFIX3, which is a bile acid metabolism disorder, for which we will initiate clinical development in 2023. We have limited competition there, very limited competition, actually. A differentiating modular platform with the modulation of neutralizing antibody, either in patients that are seropositive naturally or those that have already been exposed to the gene therapy and potentially for readministration. And as well, a non-integrative approach, which is aimed to target especially the growing organ and the pediatric population and allow to replicate the episome, the transgene, during the mitosis without being integrative. It's not gene editing. And finally, we have strong, again, manufacturing collaborations with one with Pfizer and some with other CDMO, again, allowing us to have a high yield and a very high-quality product. We have been seeing very exciting full empty ratio recently with Pfizer at high scale. So we think we are in a good shape now to deliver for the clinic. Thank you for your attention.